Welcome to the basics lecture on cell organelles. The largest organelles of the cell are labeled here. Take a moment to label your notes as well. Many cell organelles are organized around making proteins, so here are the steps of protein synthesis in a nutshell. First, instructions for a protein are coded in DNA molecules in the nucleus. This information is copied to mRNA through a process called transcription that we'll talk more about later. mRNA is processed by cutting out or removing different bits in RNA processing. And then the mRNA passes through a nuclear pore into the cytoplasm and attaches to a ribosome. The ribosome matches the pattern of the mRNA to amino acids in a process called translation, sticks the amino acids together, and makes a protein molecule. Our first organelle will be the nucleus. It contains DNA and a structure called the nucleolus. The nucleus is surrounded by its own membrane called the nuclear envelope, which is a double membrane with an outer and an inner lipid bilayer. The envelope is full of holes called nuclear pores, and they're created by integral proteins called pore complexes, shown close up here. Small molecules can diffuse through these pores, but large molecules require a nuclear import or export tag to be allowed to pass through the pore. Inside the nucleus are the cell's chromosomes. DNA molecules are wrapped around histone proteins and then coiled into a thick string. We call the diffuse mass in DNA and proteins chromatin. And if we're ever discussing one strand of DNA plus its proteins, we call that a chromosome. The nucleolus is just a darker staining region of the nucleus. It contains a lot of RNA molecules and small proteins that are being assembled into ribosome subunits. Next, the endoplasmic reticulum is a series of many flat sacs made of membrane, just like the plasma membrane. The endoplasmic reticulum is actually continuous with the nuclear envelope, as you can see here. It has two main functions that vary in different cells, and I like to call them factory functions and pocket functions. By factory, I mean that the ER is responsible for manufacturing new molecules, breaking down existing molecules. By pocket functions, I mean that many molecules or ions are kept separate from the cytoplasm by being stored inside the lumen of the ER. The smooth ER is a region of endoplasmic reticulum that doesn't have ribosomes bound to it. The factory function of this is to make lipids, like phospholipids and steroids. They also hold enzymes in place to break down carbohydrates for fuel or to break down toxins in the body to prevent cell damage. The pocket function of the smooth ER is to hold things away from the cytoplasm. In muscle cells, for instance, the smooth ER stores calcium ions. Rough ER is called rough because at any moment there are many ribosomes attached to the sacs. The factory function of the rough ER is to make membrane for the cell to use in the plasma membrane and in other organelles. The pocket function of the rough ER is to hold the beginnings of proteins. Any protein that a cell needs to put someplace other than the cytoplasm will start at the rough ER. You've noticed how I've mentioned ribosomes a lot, so let's talk about them more now. They're a complicated molecular machine made out of both protein and ribosomal RNA. You can see that in the slightly more realistic diagram here. Here's some alpha helices associated with proteins, and you can see the nucleic acids twisted around themselves there. These are assembled in the nucleolus of the nucleus and float in the cytoplasm as separate subunits, large and small. Ribosomes are found in two main locations. They can either be free in the cytoplasm, seen as little brown dots there. Some free ribosomes, once they bind to mRNA and begin protein synthesis, are then escorted to the rough ER and bind there. These are called bound ribosomes. The Golgi apparatus is another stack of membrane, not unlike the endoplasmic reticulum that we've looked at already. Each flattened sac is called a cisterna. Here's one right here. Proteins that are made in the rough ER are not yet complete. Many need to have carbohydrates or phosphate groups added or to be bound together to other proteins. The Golgi serves as a packaging center that finishes the manufacture of proteins. New proteins enter here on the cis side. They're processed within the Golgi, and then they exit on the trans side. So you can think of this as being a series of steps. Proteins begin synthesis at the rough ER, transported by vesicle to the cis phase of the Golgi. Processing, different things are done to them. They emerge from the trans phase of the Golgi. Some of them head off to become organelles like lysosomes, we'll talk about in a minute. Others of them are vesicles that are escorted to the plasma membrane, where the proteins that are made are then secreted. You should add a comment to your notes that the organelles we've talked about so far, nucleus, endoplasmic reticulum, and Golgi, together make up the endomembrane system of the cell. A lysosome is a specialized vesicle that the cell uses to digest large molecules or old organelles. It has three important components: a lipid bilayer, pumps that transport hydrogen ions or proteins into the lysosome that make it acidic inside, because high proton concentration means acidity, and enzymes that reside in the lysosome that break down molecules and help recycle them. Lysosomes are made by the endomembrane system. The proteins inside and the membrane are created by the rough ER, modified in the Golgi, and then the vesicles bud off the Golgi to float in the cytoplasm. When a lysosome is needed, by the cell bringing in some food, maybe by phagocytosis, the vesicle comes in, binds to that vesicle with molecules inside to be digested, fuses with it. The digestive enzymes are released and can break apart the molecules of food and recycle them in the cell. Okay, at this point you've been introduced to several different proteins. There are enzymes, like this one, microtubules, motor proteins, transporters, receptors in the membrane, and secreted proteins like antibodies, which are used in the immune response, or maybe insulin, which is used to control digestion. These are outside the cell. Even collagen is a molecule part of the extracellular matrix. So how is it that some proteins stay in the cytoplasm, some end up in the membrane, or some are secreted? Well, to explain this, we'll start with an insulin protein as an example. Insulin is a small protein hormone that's used in digestion. The mRNA for this protein is transcribed in the nucleus, exits the nucleus through a nuclear pore, meets up with a ribosome, and translation begins. The first 20 or so amino acids emerge from the ribosome, and they're a special series of amino acids called a signal peptide that usually has a lot of hydrophobic amino acids in it. These amino acids favorably matched with a pocket in another molecule called a signal recognition particle, or SRP, shown here. The SRP binds to the signal peptide, and this complex is then escorted to the rough ER. You can see that the SRP can bind to a pore in the rough ER called a translocation complex. And when it binds, it threads the newly forming peptide through the plasma membrane and into the lumen of the rough ER. As you can see, the first thing to go through is the signal particle. Then the rest of the polypeptide follows. There's an enzyme that breaks off the signal peptide, so it's removed, and the remaining protein is then inside the lumen of the rough ER. When translation is finished, the ribosomes fall apart, the mRNA leaves, and those diffuse away to continue on more episodes of translation until the mRNA falls apart. Meanwhile, this protein is ready to be modified more fully to be used as insulin. So now you should begin to understand that different proteins start in the cytoplasm, but if they have a signal peptide, they end up in different places inside and outside the cell. So let's do this again with 3 examples so that you have a chance to follow what happens to 3 different sorts of proteins. A secreted protein like insulin is going to have a signal peptide when translation begins. This is going to send the ribosome to the rough ER, which puts the peptide inside the rough ER. This is going to bud off in a vesicle that's going to be in the Golgi, modify the protein. It's going to be in a vesicle that leaves the Golgi, and is then sent to the plasma membrane using microtubule and the motor kinesin. The vesicle then binds with the plasma membrane, releases the protein out into the environment, and you have a secreted protein. If you need a protein to be in the membrane, like our old friend aquaporin, this also generates a signal peptide, binds to a SRP that escorts it to the rough ER, but this protein has several hydrophobic regions in it because it's a membrane protein. Because it's uncomfortable being in the lumen of the ER, it's actually threaded into the membrane of the rough ER. This then breaks off as a vesicle with the protein in the membrane, arrives at the Golgi, exits the Golgi, arrives at the plasma membrane, and now when the vesicle binds to the plasma membrane and fuses with it, the membrane protein is already built into the new membrane that's added to the plasma membrane. Our last example of a cytosolic protein, I don't know, let's do dynein. When translation begins with this ribosome and mRNA, there is no signal peptide. Because there's no signal peptide, the SRP will not bind to it, it will not be escorted to the rough ER, and the protein is just finished in the cytoplasm. That's how the cell sends proteins to different locations in the cell. I haven't forgotten about mitochondria or chloroplasts, but we'll talk about them when we discuss metabolism. So this completes our basics video. Write out the answers to the following questions. Thanks for watching.